Welcome to Nexus Clinical. Before diving into this lecture, I want to ensure we're all on the same page. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this content is at the user's risk. This is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay seeking medical advice for any medical condition they have, and they should consult their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures. The landscape of pharmaceutical management is currently undergoing a seismic shift, driven by the rapid emergence of advanced therapeutic modalities that fundamentally challenge the foundational principles of traditional formulary systems. For decades, health systems have relied on robust processes built around the predictable science and economics of small molecule drugs. This material presents the Pharmacist's Playbook for Formulary Leadership, a strategic guide to biologics, cell and gene therapies, and biosimilars. Recognizing that the rise of biologics, their biosimilar counterparts, and the revolutionary potential of cell and gene therapies, cellular gene therapies, demands a new approach. These advanced therapies are not merely incremental innovations. They represent a fundamental departure in manufacturing, complexity, cost, and clinical uncertainty. For the modern pharmacy leader, mastering this new domain is an imperative for ensuring both optimal patient outcomes and the financial solvency of the organization. This guide establishes the essential scientific and regulatory context, dissecting the fundamental differences between small molecules, biologics, biosimilars, and cellular gene therapies. It further emphasizes that the necessary strategic evolution in formulary management requires moving from simple cost comparison to a more sophisticated practice of complex clinical, operational, and financial risk management. The following learning objectives are designed to capture the core scientific, regulatory, operational, and financial competencies required for effective formulary leadership in advanced therapeutics. They reflect a strong foundation in pharmacy education. First, learners should be able to differentiate between small molecule drugs, biologics, biosimilars, and cell and gene therapies based on their fundamental differences in source, molecular size, manufacturing complexity, stability, and route of administration. Next, they should be able to explain the distinct regulatory pathways utilized by the Food and Drug Administration, specifically the Biologics license application process for novel products including cell and gene therapies, and the abbreviated pathway for biosimilars. Learners should also be capable of applying the standard that considers the entire body of evidence to critically assess the data package for a biosimilar. This includes recognizing the importance of analytical studies and pharmacokinetic and pharmacodynamic studies over redundant phase 3 clinical trials. Additionally, they should be able to distinguish between a biosimilar and an interchangeable biological product, clarifying the clinical and legal implications of each designation for pharmacist-level substitution within a health system. The objectives also include leading the formulary management process effectively. This involves ensuring that the Pharmacy and Therapeutics Committee operates objectively and expanding governance through proactive, cross-functional collaboration with finance, supply chain, and information technology teams. Developing a robust budget impact analysis methodology is another key skill. Learners should be able to create data-driven business cases for adopting biosimilars, including modeling patient populations, uptake rates, and price discounts. The role of pharmacists in the operational management of cell and gene therapies must also be understood. This includes managing the chain of identity and custody, handling specialized cryopreserved storage, and performing aseptic preparation such as thawing before infusion. Furthermore, They should be able to analyze the financial challenges posed by the high upfront costs of cell and gene therapies. They need to evaluate innovative risk-sharing payment models, such as outcomes-based rebates and contracts based on payment installments. Strategic planning skills are important as well. This involves designing and executing implementation plans that incorporate stakeholder education and information technology system integration, including updates to electronic health records and computerized physician order entry systems, to ensure a smooth transition to formulary-preferred advanced therapeutics. Finally, learners should be able to define and track key performance indicators such as biosimilar conversion rates and the comparison of realized versus projected budget impacts. These metrics help monitor accountability and demonstrate the financial and clinical value of formulary initiatives. The landscape of pharmaceutical management is experiencing a major shift, driven by the rapid rise of advanced therapeutic options that challenge the core principles of traditional formulary systems. For many years, health systems have built strong processes around the predictable science and economics of small molecule drugs. However, the emergence of biologics, the introduction of biosimilars, and the groundbreaking potential of cell and gene therapies, cellular gene therapies, require a new approach. These therapies are not just small improvements. They fundamentally change manufacturing, complexity, cost, and clinical uncertainty. For today's pharmacy leaders, understanding this new environment is essential, not optional, to ensure the best patient outcomes and the financial health of their organizations. This section provides the key scientific and regulatory background that supports all upcoming strategic formulary decisions, highlighting the main differences between these advanced therapies and traditional ones. A nuanced understanding of the unique features of each therapeutic class is essential for effective formulary planning. The shift from chemically synthesized small molecules to living cell-based therapies marks an exponential increase in complexity, which directly affects every aspect of their management, from procurement and handling to clinical use and cost considerations modeling. For most of modern pharmacy, small molecule drugs have been the standard. These are medications with low molecular weight, typically produced through predictable and reproducible chemical synthesis. Their structures are well-defined and can be fully characterized, enabling the creation of identical generic copies once patents expire. This predictability underpins traditional formulary management, where generic substitution is a simple clinical and financial decision, usually authorized at the pharmacy level without requiring review by the Pharmacy and Therapeutics Committee. Biologics represent a significant advancement in the field of medicine due to their complexity. Unlike chemically synthesized drugs, biologics are large and intricate molecules that are produced from living organisms, including human, animal, and microbial cells. This category encompasses a wide range of products such as vaccines, blood-derived products, and therapeutic proteins like monoclonal antibodies. These biologic medicines have transformed treatment options and continue to expand the possibilities for managing various health conditions. The source of biologic drugs is biological, which results in molecules that are significantly larger than those of small molecule drugs, ranging from 100 to 1,000 times larger. These molecules are composed of complex arrangements of proteins, sugars, and nucleic acids. Unlike small chemical compounds, biologics often exist as heterogeneous mixtures of closely related variants rather than as single, pure chemical entities. An illustrative example of the difference in scale between these types of drugs is the comparison between aspirin, a small molecule, and the biologic infliximab, marketed as Remicade. Infliximab contains over 6,000 carbon atoms, nearly 10,000 hydrogen atoms, and about 2,000 oxygen atoms, highlighting the substantial complexity and size of biologic molecules. The production of biological medications depends on complex, multi-stage processes that involve living cell cultures. These processes are proprietary and expensive, and they tend to fluctuate naturally. Even small changes in the manufacturing environment can lead to variations in the final product. As a result, biological medications are highly sensitive to external conditions such as heat and are vulnerable to microbial contamination. To ensure their stability and effectiveness, these medications require special handling procedures. This often includes refrigeration and maintaining a controlled temperature environment, commonly known as cold chain logistics, during storage and transportation. Clinical characteristics of biologic therapies include their method of administration and potential immunogenicity. These therapies are usually given through injection or infusion because their large size and protein-based structure prevent them from being absorbed when taken orally. Biologics often have highly targeted mechanisms of action, which can provide superior effectiveness for treating complex diseases such as cancer and autoimmune disorders. However, their biological origin also introduces a significant risk of provoking an immune response that might be unwanted and potentially harmful to the patient. A biosimilar is a biological product approved based on demonstrating that it is highly similar to an already FDA-approved biologic, known as the reference product. It is important to understand that biosimilars are not generic biologics. The natural variability in the manufacturing process makes it impossible to produce an exact copy of a complex biologic. Therefore, the regulatory standard emphasizes a high degree of similarity with no clinically meaningful differences in safety, purity, and potency. While slight differences in clinically inactive components are allowed, the active components must be structurally and functionally equivalent. Cell and gene therapies are at the forefront of biomedical innovation, providing potential one-time, curative treatments for diseases that were previously difficult to treat. The category of biological products includes items such as somatic cells, tissues, and gene therapies. These products are regulated by the Food and Drug Administration Center for Biologics Evaluation and Research. Gene therapy aims to modify the expression of a person's genes in order to treat or cure diseases. Cell therapy involves transferring live cells into a patient to achieve a therapeutic effect. An example of this type of therapy is chimeric antigen receptor T-cell therapy, which is a form of cell-based gene therapy. In this treatment, a patient's own T-cells are genetically engineered to recognize and attack cancer cells. Transformative potential and unprecedented challenges. The adoptive use of cellular gene therapies holds the promise of transforming the lives of patients. This is especially true for individuals with rare genetic disorders or advanced cancers for which no other treatments are currently available. However, realizing this potential involves significant challenges. The costs of these therapies are often extraordinary, frequently reaching into the millions of dollars per treatment. Additionally, there are numerous logistical, administrative, and ethical issues that must be addressed. These challenges strain existing healthcare infrastructure and reimbursement systems, making widespread access to these therapies complex. The journey from small molecules to cellular gene therapies is not a simple continuum. It is a ladder of increasing complexity. Each step up this ladder introduces new variables that fundamentally change the risk-benefit equation for a health system. While small molecule generics provide predictable clinical outcomes and certain financial savings, biosimilars require a new level of trust in analytical data over duplicative clinical trials. Innovator biologics for chronic diseases bring high but sustained costs that can be managed through traditional contracting. Cellular gene therapies, however, pose a different kind of challenge. A single, catastrophic cost for a treatment whose long-term value and durability are often uncertain at launch. This progression calls for a strategic evolution in formulary management, shifting from a focus on simple cost comparisons to a more complex practice of managing clinical, operational, and financial risks. The U.S. Food and Drug Administration has established separate regulatory pathways for these therapeutic classes, and a thorough understanding of these pathways is essential for any pharmacist leading a formulary review. The evidence package presented to a P&T committee directly reflects the regulatory requirements needed for approval. Therefore, understanding what the FDA required, and equally important, what it did not require, is fundamental to an evidence-based evaluation. The traditional pathway for all novel or innovator biological products, including cellular gene therapies, is the Biologics License Application, BLA, submitted under Section 351A of the Public Health Service Act. This is a comprehensive and self-contained application that must include all the necessary data to independently verify the safety, purity, and potency of the product. Typically, this involves a thorough collection of data derived from preclinical studies, which include laboratory and animal testing. Additionally, a complete set of clinical trials conducted on humans is required. These trials are generally organized into 3 phases. Phase 1, which focuses on safety. Phase 2, which evaluates efficacy and determines appropriate dosage. And large-scale Phase 3, designed to confirm the efficacy and safety of the product through extensive testing. Focusing on manufacturing processes is crucial because, in the case of biologics, the process essentially defines the product. The biologics license application places significant emphasis on chemistry, manufacturing, and controls information. The Food and Drug Administration conducts thorough inspections of manufacturing facilities to ensure that they can produce biologic products consistently and meet the highest quality standards. Minor changes in the manufacturing process can substantially affect the final product's clinical profile, making rigorous oversight essential. Recognizing that requiring full, duplicative clinical programs for follow-on biologics would be prohibitively expensive and potentially unethical, Congress created an abbreviated licensure pathway for biosimilars as part of the Biologics Price Competition and Innovation (BPCI) Act of 2009. This pathway, under Section 351(k) of the PHS Act, is designed to increase competition and lower healthcare costs by reducing the development burden for manufacturers. The core principle of the 351(k) pathway is that it allows a biosimilar manufacturer to rely, at least in part, on the Food and Drug Administration's previous determination of safety and effectiveness for the reference product. This reliance is what makes the pathway abbreviated. However, it is important to clarify that the term abbreviated does not mean a lower standard of review. Instead, it refers to a more streamlined and targeted approach to generating data, which enhances efficiency without compromising the quality of the review process. The foundational element of the biosimilar approval process is the approach known as the totality of the evidence. This approach involves a step-by-step process that assembles a thorough and complete package of data. The purpose of this data is to support the demonstration that the biosimilar product closely resembles the reference product in terms of structure, function, and clinical performance. By building this comprehensive evidence, regulators can make informed decisions about the biosimilarity of the product, ensuring safety and efficacy for patients. The process begins with extensive and highly sensitive analytical studies that compare the structure and function of the proposed biosimilar to the reference product. This comparison, often described as fingerprint-like, is the most critical part of the data package and forms the foundation of the biosimilarity assessment. If any minor differences are observed in the analytical characterization or if residual uncertainty remains, targeted animal studies and clinical pharmacology studies in humans are conducted. These studies compare how the biosimilar and the reference product behave in the body and how they affect the body, and they are highly sensitive in detecting potential clinical differences. If uncertainty persists after these steps, a final confirmation is often sought through comparative clinical studies. These studies are designed to assess the immune response and ensure there are no meaningful clinical differences in a sensitive group of patients. The main goal of this study is to confirm the similarity between the biosimilar and the reference product without reestablishing the efficacy of the molecule, which has already been demonstrated for the reference product. The Biologics Price Competition and Innovation Act established 2 categories of biosimilar products, which have significant practical implications for pharmacists and healthcare systems. A biosimilar is a product proven to be highly similar to a reference biologic, with no clinically meaningful differences. However, the designation of a biosimilar does not automatically allow a pharmacist to substitute it for the prescribed reference product. Such substitution requires approval or intervention by the prescriber. An interchangeable biologic, on the other hand, meets a higher standard that involves additional data demonstrating not only biosimilarity, but also that the product can be expected to produce the same clinical results in any given patient. Moreover, for products that are administered more than once, it must be shown that switching between the interchangeable product and the reference biologic does not pose a greater safety risk or reduce efficacy compared to using the reference product without switching. When a biologic is designated as interchangeable, it permits a pharmacist to substitute it for the reference biologic at the pharmacy level, in accordance with state laws, without needing direct approval or intervention from the prescriber. The regulatory pathway selected influences the type and amount of evidence that a P&T committee reviews. For a novel 351A product, the committee conducts a traditional, detailed evaluation of extensive clinical trial data to determine its role in therapy. For a 351K biosimilar, the committee's function shifts. It is not re-evaluating the clinical effectiveness of the reference product. Rather, it is performing a meta-assessment to confirm confidence in the FDA's comprehensive totality of the evidence review. The pharmacist leadership is essential in helping the committee understand this new evidence framework, clarifying why requiring redundant Phase 3 trials is often unnecessary and how extensive analytical data offers the scientific basis for biosimilarity. While cellular gene therapies are approved under the 351A BLA framework, their novelty and complexity have necessitated a highly specialized and evolving regulatory approach from the FDA. The Center for Biologics Evaluation and Research has provided numerous guidance documents specifically related to cell and gene therapies. These documents cover a wide range of topics, including changes in manufacturing and quality control processes, long-term follow-up of patients, and the design of clinical trials. Often, these therapies are used to treat rare diseases, which typically involve very small patient populations. This guidance aims to ensure that the development and application of cell and gene therapies are conducted safely and effectively. To foster innovation and accelerate patient access to potentially life-saving therapies, the Food and Drug Administration employs significant regulatory flexibility. This flexibility includes accepting innovative clinical trial designs, such as studies where patients serve as their own control, adaptive trial designs, and the incorporation of real-world evidence in post-approval data collection. Additionally, the agency offers expedited programs, like the Regenerative Medicine Advanced Therapy designation, which provide more intensive guidance from the Food and Drug Administration and opportunities for accelerated approval. The Food and Drug Administration has enhanced its communication efforts with sponsors, especially considering the unique challenges faced in cell and gene therapy development. To facilitate this, the agency has established new platforms for early and regular dialogue. Programs such as the Initial Targeted Engagement for Regulatory Advice on CBR products and the creation of the Office of Therapeutic Products are designed to offer expert guidance to developers well before submitting a biologics license application. These initiatives aim to streamline the development process and foster more effective collaboration between regulators and developers. For cellular gene therapies, the evidence package presented to a P&T committee is often sparse compared to traditional drugs, characterized by smaller trial sizes, non-traditional endpoints, and limited long-term data. This environment of heightened uncertainty elevates the importance of post-implementation monitoring, real-world data collection, and the negotiation of novel payment contracts that link reimbursement to sustained clinical outcomes. Armed with a foundational understanding of the science and regulation of advanced therapeutics, the pharmacy leader must then design and implement a rigorous, evidence-based, and financially sustainable formulary management process. This involves going beyond the traditional scope of the P&T committee meeting to establish a cross-functional governance structure capable of addressing the complex challenges these products pose. The pharmacist's role is not just to present clinical data, but to lead a strategic, institution-wide effort that guarantees safe, effective, and sustainable access to these groundbreaking therapies. The modern P&T committee functions as the official decision-making body for formulary management, but its effectiveness in the era of advanced therapeutics depends on the pharmacist's ability to lead a much broader, more integrated process. Success is measured not just by the decisions made in the meeting room, but by the smooth implementation of those decisions throughout the entire health system. Pharmacists are uniquely equipped to lead the assessment and incorporation of biologics, biosimilars, and cellular gene therapies. Their extensive knowledge in biopharmaceutics, clinical literature review, and medication use systems makes them natural leaders in this complex process. In this role, the pharmacist must act as an integral educator, advocate, and trailblazer, guiding the organization through unfamiliar territory with objectivity and a strong focus on patient care. To effectively adjudicate these complex therapies, the P&T committee must operate under a set of core principles championed by its pharmacy leadership. The composition of the committee should accurately represent the multidisciplinary nature of modern healthcare. It is essential to include not only physicians from relevant specialties and clinical pharmacists, but also representatives from nursing, hospital administration, finance, and billing departments. This diverse membership ensures that decision-making is comprehensive, providing a complete perspective on the therapy's impact, from bedside administration to reimbursement processes behind the scenes. The pharmacist leader serves as the guardian of an objective, evidence-based process. It is essential for them to actively work to eliminate personal biases, anecdotal experiences, and undue influence from the industry. Their focus should remain on guiding the committee's deliberations toward high-quality data sourced from reputable outlets. Such sources include peer-reviewed scientific journals, professional society guidelines, and documents issued by the Food and Drug Administration. While managing financial resources effectively is essential, the primary aim of formulary decisions should always be to enhance patient outcomes. The committee's work must be rooted in a dedication to ensuring access to the highest quality, safest, and most effective therapies. At the same time, it is important to consider the economic impact on both the individual patient and the healthcare system as a whole. The committee responsible for policies related to medication use should serve as the central authority within the organization. Its responsibilities should go beyond simply adding or removing drugs from a list. The committee must also develop and oversee all policies associated with medication use. This includes guidelines for prescribing, protocols for substitution, safety monitoring, and procedures for administration. Implementing these comprehensive policies helps ensure that the medication use process is standardized, safe, and efficient for all therapies. This is especially important for complex treatments such as biologics and cell and gene therapies. A formulary decision for a biosimilar or cellular gene therapies is not an isolated event. It is the catalyst for a cascade of operational changes throughout the health system. A forward-thinking pharmacy leader recognizes that the P&T committee is the hub of a hub-and-spoke governance model, and that proactive engagement with the spokes is essential for successful implementation. Waiting until after a P&T decision to engage other departments is a recipe for failure, leading to implementation delays, unforeseen costs, and clinical confusion. The pharmacist must act as the central liaison, orchestrating collaboration with key departments before, during, and after the formal review. Effective collaboration with finance and revenue cycle management teams is essential from the early stages and continues throughout the process. When dealing with biosimilars, it is important to work closely with these teams to develop the budget impact model, ensuring that the assumptions made are realistic and that the projected savings are consistent with the organization's overall goals. In the case of cell and gene therapies, such partnerships become even more critical. Discussions should cover the potential catastrophic budget impact, necessary capital investments, and the complexities involved in negotiating and managing sophisticated value-based payment models. This collaborative approach helps ensure financial feasibility and alignment with organizational priorities. The pharmacist must collaborate closely with leaders in the supply chain to evaluate the reliability of manufacturers, negotiate contracts, and plan for the specific logistical requirements of these drugs. This includes ensuring the integrity of the cold chain for biological products and developing specialized procedures for receiving, storing, and handling therapies, such as cellular gene therapies. These procedures may involve cryopreservation and other advanced techniques to maintain the efficacy and safety of the medications throughout the supply process. The Information Technology Department plays a vital role in implementing formulary decisions. It is essential for the pharmacist to lead efforts ensuring that electronic health records, computerized prescriber order entry systems, and billing platforms are kept up to date. This process involves adding new drugs along with their non-proprietary names and suffixes, developing order sets, and creating clinical decision support alerts to promote proper usage. Additionally, the configuration of billing systems is crucial to ensure accurate charge capture and reimbursement. Collaborating with physician champions and nursing leadership is crucial for the development of clinical guidelines, administration protocols, and educational materials. Such collaboration helps promote the adoption of these standards and ensures their safe use at the point of care. This proactive, integrated approach transforms the pharmacist's role from that of a clinical reviewer preparing a monograph for a meeting into that of a strategic program manager orchestrating a complex, system-wide change initiative. The formal P&T decision becomes a well-planned milestone within a larger, more comprehensive implementation strategy, rather than a starting point for a series of reactive and disjointed operational scrambles. Conducting a formulary review for a biosimilar requires a fundamental shift in mindset from evaluating a novel drug. The primary goal is not to re-establish the efficacy and safety of the underlying molecule, which has already been confirmed by the reference product. Instead, the focus is on critically evaluating the evidence supporting the FDA's biosimilarity determination. A structured, multi-domain framework ensures a thorough and efficient review that builds stakeholder confidence and encourages successful adoption. The entire evaluation must be based on the totality of the evidence principle. The P&T committee's review process should logically follow the FDA's own step-by-step approach, starting with the foundational analytical data and progressing through pharmacological and clinical evidence to confirm the absence of significant differences. The pharmacist's role is to educate the committee on this framework, guiding the discussion away from searching for large, redundant clinical trials and toward understanding the scientific rigor behind the biosimilar approval pathway. A frequent mistake in biosimilar reviews is an over-clinicalization of the process, where committees get stuck debating minor, clinically insignificant analytical differences while ignoring the essential institutional and operational planning needed for successful implementation. A strategic pharmacy leader will maintain balance in the discussion, ensuring that as much effort is put into developing the education plan, IT strategy, and substitution policies as into reviewing the clinical data. This approach acknowledges that the main barriers to achieving biosimilar savings are often operational and educational, rather than clinical. This framework is designed as a checklist to serve as a practical tool for pharmacists, guiding their pharmacy and therapeutics committee through a comprehensive and standardized evaluation of biosimilars. The first domain focuses on assessing clinical parameters. Pharmacists should review the manufacturer's scientific justification for the extrapolation of indications, ensuring they understand the rationale behind extending use to different patient populations. It is also important to examine data comparing pharmacokinetics and pharmacodynamics profiles to support similar exposure and response levels between the biosimilar and the reference product. Confirming that there are no clinically meaningful differences in efficacy and safety outcomes is essential. Additionally, a critical review of comparative immunogenicity data should be conducted to ensure there is no increased risk of immune response compared to the original reference product. The second domain focuses on product and logistical assessment. It involves identifying the proper non-proprietary name of the biosimilar, including the unique four-letter suffix, and planning its integration into all information technology systems. Additionally, it requires assessing the credibility and manufacturing experience of the sponsor, as well as evaluating the reliability of the supply chain to minimize the risk of shortages. It is also important to identify and evaluate the clinical and operational impacts of differences in formulation, concentration, or the administration device, such as an auto-injector. The third domain pertains to institutional and economic assessment. One key aspect is confirming the interchangeable status of the biosimilar by consulting the Food and Drug Administration's Purple Book, which influences substitution policies. Developing a clear institutional policy is essential. This policy may permit automatic substitution if the biosimilar is deemed interchangeable or may involve a therapeutic interchange protocol if it is not. Furthermore, policies should be established for the transition of care, addressing both patients who are newly starting treatment and those who are stable on the reference product. An educational component is also critical. A proactive plan should be developed to educate prescribers, nurses, and patients. This plan can include utilizing physician champions and explaining concepts such as the totality of the evidence to ensure understanding and acceptance. The arrival of cell and gene therapies requires a fundamental rethinking of the purpose and structure of the formulary review process. These therapies, with their potential for cure, multi-million-dollar price tags, and high operational complexity, cannot be adequately evaluated through a traditional P&T committee. The formulary decision for cellular and gene therapies is not just a clinical choice. It represents a significant institutional strategic commitment to invest in and support an entirely new service line, one that involves substantial financial risk and the development of new operational infrastructure. Recognizing that the scope of evaluating cellular gene therapies goes well beyond just clinical effectiveness, leading health systems are creating specialized, emerging therapies or advanced therapeutics committees. Unlike a traditional P&T committee, which may have limited representation from the C-suite, these specialized bodies include senior leaders from finance, contracting, legal, operations, and clinical medicine from the very beginning of the review process. This structure ensures that the decision to adopt a cellular gene therapy is fully informed, comprehensive, and strategic, with financial and operational feasibility given equal importance to clinical merit from the start. The hospital pharmacist is the indispensable linchpin in the entire cellular gene therapies process, particularly for complex autologous therapies like CAR-T. Their responsibilities span the full continuum of care, transforming their role from medication expert to service line co-developer. Governance, logistics, and chain of custody are critical aspects in the management of cellular gene therapies, which are regulated as medicinal products. The pharmacist holds both legal and professional responsibility for overseeing these processes. This involves managing the entire chain of identity and chain of custody, which constitutes a highly complex logistical undertaking. The process begins with ordering and procurement, where the pharmacist coordinates the intricate process of placing orders with the manufacturer. Upon receipt, the cells are cryopreserved, and verification is essential. This includes checking the product's integrity, labels, and temperature monitoring data to ensure quality and safety. Storage is another vital component, requiring specialized liquid nitrogen freezers and meticulous record-keeping to ensure accurate inventory management and traceability. When it is time to administer the therapy, the pharmacist develops and follows validated, aseptic procedures for thawing the product. This step must be executed immediately prior to administration, as errors during this stage can result in the loss of the entire dose, which may be worth several $1 million. Clinical management and patient safety are critical aspects of the healthcare process, with the pharmacist playing an essential role in managing the patient throughout the treatment journey. Before treatment begins, the pharmacist is responsible for preparing and dispensing the chemotherapy regimens known as bridging and lymphodepleting therapies, which patients receive prior to the infusion of cellular gene therapies. The pharmacist also leads the proactive management of severe and unique toxicities associated with these treatments. This includes developing institutional protocols and ensuring the immediate availability of rescue medications such as tocilizumab and corticosteroids. These measures comply with federal safety programs that aim to mitigate potential risks. Additionally, providing comprehensive education to patients and their caregivers is vital. This education covers the entire treatment process and highlights the signs and symptoms of possible toxicities, enabling early recognition and prompt intervention. System readiness and infrastructure development are crucial components in the safe delivery of cellular gene therapies. Pharmacists play a vital role as the key architects of the institutional infrastructure needed for this process. This involves building electronic health record order sets specifically for chemotherapy and toxicity management. Additionally, they develop standardized operating procedures for each step of the logistical chain. Training nursing staff, pharmacy personnel, and medical staff on these complex protocols is also essential to ensure safety and efficacy. The evaluation process for cellular gene therapies must address a unique set of questions that go far beyond those typically considered for drug monographs. One of the key challenges is assessing the clinical evidence, which is often based on smaller, single-arm clinical trials. This is due to the rarity of the diseases these therapies aim to treat. As a result, the review process must focus on evaluating both the magnitude and, critically, the durability of the observed clinical benefits. The committee responsible for the evaluation must be comfortable making high-stakes decisions based on limited long-term data. Additionally, they They need to establish a robust plan for tracking the outcomes of these therapies in real-world settings after they have been administered. The central question for the committee is not merely whether this therapy is effective. Instead, they must consider whether the institution can safely, effectively, and reliably deliver the treatment. To address this, a comprehensive assessment of institutional readiness is necessary. This assessment should evaluate staffing levels and expertise, including specialized nurses, neurologists, and intensivists. It should also examine facility capabilities such as the availability of apheresis centers, cell processing laboratories, and intensive care unit beds. Additionally, the assessment must consider the complex logistical pathways required to provide the therapy. This thorough evaluation ensures that the institution is prepared to implement the treatment safely and efficiently. The evaluation of clinical therapies cannot be separated from financial considerations. It is essential to assess the therapy within the context of its significant cost and to consider any innovative payment models proposed by the manufacturer, such as contracts based on the value delivered. The participation of the institution's contracting and finance executives in the decision-making process is crucial and must be actively and directly involved. The pharmacist, with a unique vantage point across the clinical, operational, and financial domains of cellular gene therapies delivery, is the critical integrator of these diverse considerations. Their leadership is essential to prevent a premature clinical approval based on promising but limited data, and instead to guide the organization through a rigorous, holistic assessment. This ensures that a decision to adopt the cellular gene therapies is a fully resourced, institution-wide strategic commitment, not just an addition to the formulary list. A key element of a pharmacist's leadership in formulary management is the ability to convert clinical decisions into clear, defendable financial projections. For high-cost therapies, especially biosimilars and cellular gene therapies, a thorough financial analysis is not just supporting documentation. It is a vital part of the decision-making process itself. This section offers a detailed, step-by-step guide to measuring financial impact, progressing from the straightforward, predictable savings calculations for biosimilars to the more complex risk-sharing models needed for cell and gene therapies. To effectively communicate with finance departments and executive leadership, pharmacists must be conversant in the core principles of health economic evaluation. A budget impact analysis is the primary tool used for formulary decision-making from the perspective of a health system or payer. Its main purpose is to estimate the financial consequences of adopting a new health technology on a specific budget over a defined period, which is typically short-term, such as 3 to 5 years. The analysis helps in understanding the net change in spending that would occur if a particular drug is added to the formulary. This information is essential for effective forecasting and financial planning. Cost-effectiveness analysis compares the relative costs and health outcomes of 2 or more interventions. These health outcomes may include measures such as years of life gained or quality-adjusted life years. The results of a cost-effectiveness analysis are often expressed as an incremental cost-effectiveness ratio, which indicates the additional cost required for an additional unit of health benefit. While a cost-effectiveness analysis helps determine whether a drug offers good value for money, a budget impact analysis assesses whether that drug is affordable within the current budget. For making formulary decisions, the budget impact analysis is often more practical and influential. A well-constructed BAM is the pharmacy leader's most powerful tool for making the business case for biosimilar adoption. The process of building the model is as valuable as its output, as it forces a data-driven, cross-functional dialogue that aligns the organization around a common set of assumptions and goals. The following step-by-step methodology, based on established best practices, provides a practical guide for developing a robust BAM. The first step involves defining the structure and perspective of the model. It is essential to clearly state the perspective of the analysis, such as evaluating a hospital's drug budget, a commercial health plan, or a Medicare plan. This perspective determines which costs are relevant. For example, a hospital model would focus on drug acquisition costs, whereas a health plan might include drug costs along with administration and other medical expenses. Next, select an appropriate time horizon that is sufficiently long to capture the evolution of the market, typically 3 to 5 years. In basic economic evaluations like budget impact analyses, discounting of future costs is generally not necessary due to the short time frame. The core logic of the model involves comparing two distinct scenarios. The first is the reference scenario, often termed the world without biosimilar, which represents the current state. This scenario models the total costs, assuming only the reference biologic and any other existing treatments are used throughout the time horizon. The second scenario, referred to as the world with biosimilar, models the total costs, assuming a biosimilar is added to the formulary and gradually gains market share from the reference product over the same period. To estimate the eligible patient population, you need to determine the number of patients who would qualify for the therapy based on the relevant medical indications. This process can be approached in two ways: the top-down method and the bottom-up method. The top-down approach involves starting with the total number of individuals covered by a health plan, such as a hypothetical plan with 1 million members. Epidemiological data, including disease prevalence and incidence rates, are then applied to estimate how many of these individuals are eligible for the therapy. Alternatively, the bottom-up method is more suited to hospitals or health systems. It utilizes historical, administrative, or pharmacy claims data to identify the exact number of patients who were treated with the reference product in the past year. This approach often provides greater accuracy for institutional budgeting purposes. Once the current eligible population has been identified, it is important to project how this population might grow over the relevant time horizon. Factors such as changes in disease incidence, as well as fluctuations in the health plan's membership or the hospital's patient volume, should be considered to make an accurate projection. This step is critical and requires collaboration with other departments to ensure the data is accurate and the assumptions are realistic. All assumptions should be clearly listed and justified. The market share and uptake rates are the most sensitive assumptions in the model. In the scenario titled World with Biosimilar, it is essential to define the projected shift in market share from the original reference product to the biosimilar for each year. These projections can be informed by various factors. Historically, data from previous biosimilar launches in the United States can serve as a reference point. For instance, a conservative estimate might assume a 10% uptake in the first year, 15% in the second year, and 20% in the third year, as was used in a model for natalizumab. Additionally, institutional goals and strategies play a significant role. A proactive approach, for example, might aim for a 50% conversion rate in the first year. Furthermore, the status of the payer formulary is a crucial factor, as it greatly influences prescribing behavior. The acquisition cost of a drug typically starts with the wholesale acquisition cost, but for a more accurate estimate, it is preferable to use the actual acquisition cost after accounting for all discounts and rebates. When it comes to biosimilar drugs, it is common to assume a price discount relative to the reference product's wholesale acquisition cost. This discount generally ranges from 15 to 30% at the time of launch, but it can increase as more competitors enter the market. Additionally, a sophisticated pricing model should consider the dynamic impact of competition. This includes the possibility that the manufacturer of the original or reference product may lower its price in response to the introduction of biosimilars to remain competitive. Associated healthcare costs encompass various expenses that may differ between different scenarios. These costs can include the costs associated with administering drugs, such as the time required for infusion and the labor of nursing staff. Routine monitoring costs, such as laboratory tests, are also relevant. Additionally, the expenses involved in managing adverse events should be considered. In most analyses involving biosimilar medications, where clinical outcomes are assumed to be identical, these costs tend to be similar across different scenarios. As a result, they may not significantly influence the overall net cost outcome. Step 4 involves calculating the budget impact. For each year and scenario, the total annual cost should be determined by multiplying the number of patients using each drug by the total annual cost per patient for that particular drug. The net budget impact for each year is then calculated by subtracting the total cost of the scenario where biosimilars are not used from the total cost of the scenario where biosimilars are implemented. A negative value indicates cost savings. It is important to present these results clearly and in various formats to effectively communicate with different stakeholders. One way to do this is by showing the cumulative savings over the entire time horizon, which could be 3 or 5 years. Another useful metric is the savings per member per month, a common measure for health plans, which is calculated by dividing the total savings by the number of plan members and the number of months in the period. Additionally, the savings per treated patient provides a meaningful measure of the impact. It is obtained by dividing the total savings by the number of patients receiving treatment. This metric can be particularly effective in illustrating the financial benefits of biosimilar use. Step 5 involves conducting a sensitivity analysis, which is essential because the building information model is based on various assumptions. This analysis tests the robustness of the results by examining how changes in key input parameters affect the overall outcomes. A one-way sensitivity analysis systematically varies each key parameter individually, often by approximately 25%, while keeping all other variables constant. This process helps identify the most influential factors driving the model's results, such as the rate at which biosimilars are adopted and the discount applied to their prices. The findings from this analysis are usually presented in a tornado diagram, which visually highlights the parameters with the greatest impact. Additionally, scenario analysis is conducted to explore different potential outcomes under various assumptions. For example, scenarios may include a base case with moderate biosimilar uptake, a conservative case with low uptake, and an optimistic case with high uptake. Presenting these diverse scenarios provides leadership with a range of possible financial results, aiding in better decision-making. The BAM serves as more than just a calculation. It is a strategic communication tool. It transforms a general proposal, "We should adopt this biosimilar to save money," into a specific, measurable, and actionable business case. If we can achieve a 30% conversion rate in year 1 through targeted provider education and a preferred formulary status, we will realize a net savings of $2.5 million, which can be reallocated to support our new oncology service line. This level of specificity builds credibility and provides a clear benchmark against which the pharmacy's performance can be measured post-implementation. The financial challenge posed by cell and gene therapies is fundamentally different from that of traditional drugs or even high-cost biologics. Their potentially curative nature is coupled with an unprecedented upfront cost, creating a mismatch between the timing of payment and the realization of value. This dynamic breaks traditional reimbursement models and requires pharmacy leaders to engage in a new form of financial analysis focused on strategic risk management and the evaluation of innovative payment structures. The current reimbursement mechanisms, which are primarily based on standard fee-for-service and buy-and-bill models, are ill-suited for cellular gene therapies. This is due to several interrelated factors. One major issue is the extreme concentration of costs. Often, these therapies cost millions of dollars up front, providing benefits that may last for years or even a lifetime. Such a high initial payment creates a significant and unpredictable budget shock for payers and health systems, making it nearly impossible to manage without specific financial planning. Additionally, there is considerable uncertainty about the long-term effectiveness of these therapies at the time of their launch. Under traditional payment models, the payer or provider bears the full financial risk if the treatment fails or if its effectiveness diminishes over time. Furthermore, the structure of the United States healthcare system, which is highly fragmented, complicates matters further. Patients often change insurance plans, which means a payer might cover the substantial cost of a gene therapy for a patient who then switches to a different insurance provider the following year. In such cases, the initial payer bears all the costs, while the subsequent payer benefits from a healthier, less costly member. This misalignment of incentives discourages payers from readily covering these innovative therapies, despite their potential benefits. To address the challenges faced by the healthcare industry, manufacturers and payers are exploring various alternative and value-based payment models. These models aim to better synchronize the timing of payments with the demonstration of clinical value and to share the financial risks associated with treatment failures. It is essential for pharmacist leaders to understand the mechanics and implications of these models. One such approach is the annuity-based or staggered payment model. This model helps mitigate the impact of large, upfront costs by allowing payers to pay for therapies in installments over a predetermined period, often spanning several years. This approach transforms a potentially catastrophic single expenditure into a series of predictable and manageable payments. For example, a pharmaceutical company might offer a 5-year payment plan for a gene therapy product. Another model is the outcomes-based rebate, also known as pay-for-performance. In this case, the payer pays the full cost of the therapy upfront, but enters into a contractual agreement with the manufacturer. The agreement specifies certain clinical outcomes that must be achieved or maintained. If these outcomes are not met, the manufacturer provides a rebate to the payer, effectively sharing the risk of clinical failure. Notable instances include a company that ties rebates to both short-term measures, such as efficacy within 30 to 90 days, and long-term outcomes, like efficacy over 30 months, based on objective testing. Similarly, a different company offers to reimburse up to 80% of the therapy's cost if the patient fails to achieve and maintain transfusion independence for up to 2 years after treatment. A hybrid approach combines elements of the previous two models and is known as outcomes-based annuity. In this model, payments are made over time in installments, but each installment is contingent upon the patient continuing to meet certain predefined clinical milestones. This approach aligns payments most closely with the sustained value provided by the therapy, although it tends to be the most complex to administer. The role of the pharmacist in this evolving landscape has shifted from being primarily a cost analyst to taking on the roles of a strategic risk manager and a clinical translator. They serve as a vital link between the clinical evidence and the complex financial agreements that are now common in healthcare. This involves several key activities. First, pharmacists collaborate closely with the institution's finance and legal experts to evaluate the terms of proposed value-based contracts. Their clinical expertise is essential in helping to define outcomes that are meaningful from a medical perspective, objectively measurable, and feasible to track over time. Additionally, they develop financial models that project the institution's financial exposure under various scenarios, such as success or failure of treatment within a value-based contract. These models enable leadership to compare different payment options and assess the associated risk profiles. The conversation is no longer simply about the price of a drug, but about the structure of a multi-year financial risk-sharing agreement. The pharmacist's ability to critically appraise the clinical data and help estimate the probability of achieving the contracted outcomes is an indispensable input for the finance team's risk assessment. They are not just evaluating a therapy, they are helping to design and de-risk a major strategic and financial commitment for the organization. A meticulously researched formulary decision and a sophisticated financial model are rendered meaningless if the decision is not implemented effectively and its impact is not rigorously measured. The post-decision phase is where strategy meets reality, and it is a critical period that demands sustained leadership from the pharmacy department. This final section provides a playbook for executing formulary changes for advanced therapeutics and establishing a data-driven feedback loop to monitor performance, ensure accountability, and demonstrate value to the organization. Successful implementation is a change management exercise that requires proactive planning, clear communication, and deep cross-functional collaboration. The seamless integration of pharmacy, supply chain management, and finance or revenue cycle management forms the operational backbone of any successful formulary initiative. The pharmacy department leads the clinical aspects by developing educational materials, clinical guidelines, and order sets. They serve as the primary point of contact for prescribers and nurses. The supply chain management team handles the entire procurement-to-payment lifecycle, including contract negotiations, ordering, inventory management, and ensuring logistical requirements such as cold chain logistics are met. The finance or revenue cycle management team oversees the financial components, ensuring accurate billing, coding, and reimbursement processes. They also track the financial performance of the initiative against projections outlined in the budget impact model. Regular, structured meetings between these three departments are essential. These meetings help anticipate challenges, resolve issues promptly, and maintain alignment throughout the implementation process. One of the most significant barriers to the adoption of new formulary policies, especially for biosimilars, Is stakeholder hesitancy rooted in a lack of understanding or confidence? A robust education plan is not an optional add-on. It is a core component of the implementation strategy. Addressing provider hesitancy. Physicians often have concerns about biosimilars, which typically arise from misunderstandings of the comprehensive evidence required for approval or hesitations to switch a patient who is stable on a reference product. To overcome this, a pharmacy leader should develop a comprehensive educational campaign. This campaign should include recruiting respected clinical leaders to act as peer educators, as this approach has proven more effective than top-down administrative mandates. Additionally, the educational materials should be clear and concise, such as presentations during grand rounds, newsletters, and one-page summaries. These materials should explain the science behind biosimilarity, review the rigorous evaluation process conducted by the institution, and transparently communicate the financial benefits to the health system, which can then be reinvested into patient care. Patients may have questions or concerns about receiving biosimilar medications or new cellular gene therapies. It is important for the pharmacy to take the lead in developing educational materials that are easy for patients to understand. These materials should explain what these therapies are, why the healthcare institution has chosen to use them, and what patients can expect during treatment. Providing clear and accessible information helps manage patient expectations and build trust in the treatment process. The successful introduction of a new formulary medication depends on the precise and prompt updating of the healthcare system's information technology infrastructure. The electronic health record system must be revised to include the new medication, ensuring the correct non-proprietary name and suffix are used to prevent medication errors. Clinical decision support tools, such as alerts for best practices or setting the preferred medication as the default in order sets, should be implemented to gently guide healthcare providers toward choosing the formulary-approved option. Furthermore, all related systems, including medication dispensing cabinets, inventory management software, and billing platforms, need to be configured to recognize, track, and accurately charge for the new product. This is essential for maintaining accurate data on medication utilization, costs, and reimbursements. To ensure accountability and demonstrate the value of formulary management initiatives, a robust system for monitoring performance and reporting results to leadership is essential. This creates a data-driven feedback loop for continuous improvement. A balanced scorecard of KPIs should be developed to provide a holistic view of the initiative's success across financial, clinical, and operational domains. The biosimilar uptake or conversion rate percentage is the most crucial metric for monitoring the success of adoption. It indicates the proportion of the drug's total usage that has shifted from the original reference product to the preferred biosimilar. Tracking the total drug spend and the per member per month costs helps assess the direct financial impact on the therapeutic class. A decrease in the cost per unit or in the per member per month expenses signifies financial success. Additionally, comparing the realized budget impact to the projected budget impact provides insight into how well the initiative performs against forecasts made in the budget impact model. This comparison serves as a powerful tool for demonstrating the pharmacy's capacity to meet its financial commitments. Clinical and safety key performance indicators are essential for effective healthcare management. For medications, particularly those used in chronic therapies, it is crucial to monitor adherence rates. Ensuring patients follow their prescribed treatment regimens is vital, especially when switching to a biosimilar, as this change should not negatively affect their compliance. Additionally, tracking adverse drug events is a key component of pharmacovigilance. Maintaining a robust program to identify and record any adverse events, especially those associated with switching medications, helps monitor the safety profile in real-world conditions and enhances clinical confidence in the medication. For therapies involving cellular gene treatments, especially under value-based contracts, it is important to meticulously track clinical outcomes as defined in contractual agreements. These outcomes may include measures such as independence from transfusions and durable responses in cancer treatment. Consistent monitoring of these outcomes ensures that the therapeutic benefits are achieved and maintained over time. The operational key performance indicators include the provider formulary adherence rate, which measures the percentage of new prescriptions written for the preferred formulary agent. This indicator provides valuable insight into the effectiveness of educational efforts aimed at promoting formulary compliance. Additionally, the organization monitors dispensing errors and information technology system glitches. Tracking these issues is essential to ensure patient safety and maintain the integrity of the system during its rollout. Overall, these metrics are critical for assessing the success and safety of operational processes. The data from these KPIs should be consolidated into a clear, visual, and regularly updated performance dashboard for review by the P&T committee, department chiefs, and executive leadership. This dashboard is more than a simple report card. It is an active change management tool. By making performance transparent and public, it promotes accountability. When stakeholders can see progress toward a shared goal, it emphasizes the importance of the initiative. Additionally, the data can help identify areas of success and challenge. For example, if the dashboard shows high biosimilar adoption in the oncology department but low adoption in rheumatology, it provides the pharmacy leader with actionable data to engage the rheumatology chief, understand their specific barriers, and implement targeted interventions. This data-driven approach turns a one-time formulary decision into an ongoing performance improvement effort, ensuring the strategic and financial goals of the decision are ultimately met. The rise of biologics, biosimilars, and cell and gene therapies marks a transformative era in medicine, offering both unprecedented treatment opportunities and significant challenges to traditional healthcare models. For pharmacy leaders within health systems and managed care organizations, navigating this complex landscape requires a major shift in their role, from primarily being a clinical expert and cost manager to becoming a strategic program leader, risk manager, and cross-functional coordinator. The effective management of this new portfolio of therapies depends on several key principles outlined in this report. First, it is essential to develop a deep, nuanced understanding of the scientific and regulatory differences that set these products apart. A pharmacist cannot effectively lead a P&T committee discussion on biosimilar extrapolation or the durability of cellular gene therapies without first understanding the intricacies of the 351(k) and 351(a) approval pathways. Second, the formulary management process itself must be redesigned. It should go beyond the traditional P&T committee meetings to become a continuous, proactive governance model that involves finance, supply chain, IT, and clinical operations from the earliest review stages. For biosimilars, this means shifting the focus from repeated clinical debate to thorough implementation planning. For cellular gene therapies, it entails establishing specialized, high-level committees capable of making institutional strategic commitments. Third, financial analysis must advance from simple cost comparisons to sophisticated financial modeling and risk management. The budget impact model is a vital tool for developing a data-driven business case for biosimilar adoption, aligning the organization with clear financial goals. For cellular gene therapies, pharmacists need to become key partners in evaluating and operationalizing complex, value-based contracts that share risk and connect payment to long-term clinical outcomes. Finally, a formulary decision is just the beginning. Long-term success depends on a carefully planned implementation, continuous stakeholder education to address barriers to adoption, and a data-driven follow-up system. Using KPIs and performance dashboards turns a static decision into a dynamic performance improvement process, fostering accountability and ensuring the full realization of clinical and financial benefits. Pharmacists are uniquely positioned at the intersection of clinical, operational, and financial factors shaping this new therapeutic landscape. By embracing this expanded leadership role, as educators, strategists, and integrators, pharmacists can help their organizations leverage these advanced therapies effectively, ensuring sustainable access to innovation while maintaining high standards of patient care and financial stewardship. This was an episode of the Nexus Clinical Podcast. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this content is at the user's risk. This is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay seeking medical advice for any medical condition they have. and they should consult their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.